Hey there, doctors. Today on MedMadness, we are unlocking the secrets of bacterial structure and virulence. Why does one bug quietly colonize your throat while other sends you straight to the ICU? It's all in the structure. From capsules to toxins, let's explore the weapons bacteria use and how you can outsmart them on the USMLE step 1. Let's kick things off with the bacterial capsule. Think of it like a slick raincoat made of polysaccharides, except in Bacillus anthracis, which oddly uses a protein coat made of poly-D-glutamate. This capsule helps bacteria dodge the immune system by blocking phagocytosis. You will detect it using the Q-lung reaction or India ink, and here is the clinical kicker. If your patient has no spleen, like someone with sickle cell disease, encapsulated bugs can be deadly. So we vaccinate against them. Think Streptococcus pneumoniae, Haemophilus influenza type B, and Neisseria meningitis. Next up is pili. These tiny hair-like appendages help the bacteria stick to surfaces like mucosal membranes. Neisseria gonorrhea uses pili to grab onto the genitourinary tract like a velcro. They are also used in the horizontal gene transfers, especially the sex pilus in conjugation. And that's right, bacteria can flirt and share the DNA. Then there is flagella, the propellers. Bacteria with flagella can move whether they have a bunch all over, like say peritrichus, or just at one end, say a polar. On a test, remember that flagella are used in H antigen typing. Fine. Moving on to plasmids, they are tiny rings of DNA floating outside the chromosomes. They often carry genes for antibiotic resistance or toxins, and yes, they are transferable. That's why resistance can spread so fast in hospitals. Now let's talk about spores. These are survival bunkers. When the environment gets rough, think heat, radiation, or lack of nutrients, bacteria like Bacillus and Clostridium hunker down and form the spores. You cannot kill them with alcohol. Fine, time to face the toxins. Exotoxins are secreted proteins like deadly little syringes, while endotoxins are part of outer membrane of gram-negative bacteria. Exotoxins are specific and powerful. They might paralyze you or mess with your gut. They are also heat labile and can be used in vaccines. Endotoxins, on the other hand, come from lipid A in LPS. They are released during bacterial lysis and trigger a tsunami of cytokines. TNF alpha, interleukin 1, interleukin 6, leading to fever, hypotension, and DIC. Now, let's dive into A-B toxins. These are split into two parts. B binds to the cell and A does the damage. Some famous villains, do you remember? Diphtheria toxins blocks EF2, that is the elongation factor 2, and causes a nasty pseudomembrane in the throat. Cholera toxins ramps up cyclic AMP to cause watery diarrhea. Botulinum toxin stops acetylcholine release, causing flaccid paralysis, while tetanus toxin blocks the inhibitory neurotransmitters like GABA, causing spastic paralysis. You know about pertussis toxin? It disables GI protein, leading to lymphocytosis and that infamous whooping cough. What about superantigens? They are even sneakier. Instead of targeting a specific pathway, they light the entire immune system on fire. They bind MHC class 2 and T cell receptors simultaneously, releasing a massive cytokine storm. You know the culprits? TSST1 from Staph aureus and SPEA from Streptococcus pyogenes. The result? Toxic shock syndrome. high fever, rashes, hypotension, and potential multi-organ failure. Last but not the least, let's wrap with a deeper look at endotoxins. The toxic part of LPS, the lipid A, binds to the toll-like receptor 4. The TLR4 receptors on macrophages, triggering TNF alpha, interleukin 1 and interleukin 6, and also nitric oxides. Together, these lead to vasodilation, hypotension, and fever, possibly DIC. For your memory, just say endotoxin stands for edema, nitric oxides, DIC or death, outer membrane, TNF alpha, O antigen, extremely heat stable, interleukin 1 or 6, neutrophil chemotaxis. I hope this mnemonic helps you. Okay, a quick review. Capsules protect from phagocytosis, especially dangerous in asplenia. Pili help bacteria stick and share the DNA. Flagella for movement. Plasmids for gene smuggling. Spores for survival mode. And remember, exotoxins are potent and specific, so some are vaccine targets. Endotoxins causes generalized cause. Think of DIC and fever. AB toxins work like a two-part sabotage system. Superantigens trigger a full body immune meltdown. That's a lot of virulence, but now you are immune to confusions. Remember, don't just memorize. Understand how these structures link to clinical symptoms and therapy. You are building your diagnostic instincts one microbe at a time. Keep going, keep grinding, and let MedMadness make microbiology manageable. You have got this. Let's keep crushing it one episode at a time. And this is Dr. Ram here from MedMadness. I'll see you next episode. Thank you so much.